to have Ingrid Parker, who's a professor of ecology and in evolutionary biology at UC Santa Cruz. She's also faculty director for the Norris Center for Natural History. And her studies include plant ecology, biology of species invasions, and disease ecology. Ingrid is a fellow of the Ecological Society of America, fellow of the California Academy of Sciences. She received the UCSC Committee on Teaching Excellence in Teaching Award. She achieved the Chancellor's Achievement Award for Diversity. She was a Fulbright Fellow and the first recipient of the Jean Langenheim Endowed Chair in Plant Ecology and Evolution at UCSC. So her talk today is Worlds Within Worlds, Complex Networks of Plants and Fungi in Our Backyard.
Yeah. Thank you. Now we have to make sure that the technology is.
Does this go somewhere? I just can't.
Okay, we're good.
We turned on the.
Oh, right. No.
Right. Okay, so it goes. Yeah. And
One more step. Great. Hi, everyone. It is such a pleasure to be here, and it's so fun to see so many old friends. My smile muscles are already tired. So thank you so much for coming. No, not. Okay, we might have done this order of operations wrong, Jerry. It won't work. Let's try that again.
what, in terms of advancing?
There we go. Okay, yay. So Eli mentioned that I have a new hat now. In addition to being a faculty member, I'm the faculty director at the Norris Center for Natural History. And I wanted to make sure to say a little bit about that to make sure that everyone knows about us at the Norris Center. Our center was named after esteemed faculty member Ken Norris, who had such a big influence on so many people, including myself. And we are the hub for natural history on campus. We're very interdisciplinary. We house all of the natural history museum collections for the campus, but we also run a whole bunch of student internships. We are involved in natural history courses of all different kinds. We have a whole art and science program. I have a co-faculty director, Carolina Karlik, who's a faculty member in the art department, and she oversees all these awesome art and science programming that we have. And then we also have a friends group, the Friends of the Norris Center. Part of our mission is to bring together students who love being out in nature together with community members who love to be out in nature. And so I hope any of you who are interested in being part of that community might sign up to be a part of the Friends. And we have big annual events, including last year, our big annual symposium, which was all focused around the history of the Environmental Studies Department and Dick Cooley and Ken Norris. And so we do a lot of sort of celebrating our history as well. And then the other thing I want to say before I jump in is just I'm a huge believer in the importance of Emeriti in our community of the university. Emeriti have been really important to me throughout my career, and I just want to, and several people are, I'm so excited you're in this room. I just feel that the Emeriti play such an important role And that was part of why I served for several years on the Committee of Emeriti Relations together with Judith and Linda and Murray, and people like Barry and Lincoln and James have been really important to me as wise voices in my surroundings. And so thank you for all you do. And I hope that you really feel like you are a part of the community. And I would be remiss if I didn't really call out one particular emerita who completely changed my career, and that was Jean Langenheim. Jean Langenheim had retired 8 years before I started here, and I was hired as the first plant ecologist on campus after Jean's retirement. And she was so important to me as a mentor and supported my career in every possible way you can imagine. And I really miss her. And it was also a highlight of my career to have the opportunity to put on or support the celebration of the naming of the greenhouses on the roof of Tiemann Labs for Jean. At that event, it was so cool because we brought back Jean's former students, both undergrad and grads came back, as well as current students and many faculty from different departments and alumni and staff. So that was a highlight, and I just want to say thank you, Jean. All right, today I'm going to be telling a story that has 3 different parts. The first one is about fungi that live inside of roots. So we're going to talk about mycorrhizae and forest regeneration in the Pacific Northwest, which is some of my older work. Then we'll talk about fungi that live inside of leaves. And at first, I'm going to tell you a story about clovers, very awesome plants, California native plants, and their endophytes, so the fungi that live inside of those leaves, and their shifting roles. And then the last part of this story will be about whole networks of plants and fungi across entire communities of plants. And that is recent work that has been here at the campus, so that's where the backyard part comes in. And that work isn't even published yet, so that'll be some of the most recent stuff. So, but let's start with fungi that live inside of roots. Oh, and before I go on, actually, I should say one other thing, which is I got to be a guest at one of these Emeriti luncheons last year, and it really made a big impression on me in the Q&A period how engaged everyone was and how excited they were to have conversations about the topic. And so what we're going to do today is there'll be a little bit of a side moment here after we talk about mycorrhizae, where I'm going to give you the opportunity to talk to each other about a topic that is not really my expertise, but I think it's a lot of interest to a lot of people. So we're going to be pulling away. We're going to be stealing time from the Q&A for that. And I hope it's very experimental. So I hope you have some fun. All right. I first started working on mycorrhizae, not because I was interested in fungi per se, but because I was interested in forest regeneration and restoration. And I was contacted by a group of foresters up on Joint Base Lewis McChord, which was back then was Fort Lewis. They were doing sustainable forestry there, and they were dealing with a problem of some historical clear cuts that had been created back in the 60s, 70s, and 80s, back before people knew better, and that they could not get to revert to forest. So we were trying to understand how to get tree seedlings to grow. And in particular, my expertise was relevant because during that time period, the 60s, 70s, and 80s, what was changing was these systems had been invaded by Scotch broom, an invasive shrub, which if you've ever been up in the Northwest is really an impressive species that has kind of taken over everything. It's from Europe. In these clear cuts that have been invaded by Scotch broom, there's really nothing there but Scotch broom. So here you can see my colleague Karen Halvensack crawling through a Scotch broom thicket. It's just pure Scotch broom with tons of leaf litter on the ground and nothing else is growing there. So we set up an experiment to look at the competition between Douglas fir seedlings and Scotch broom. We planted over 8,000 trees, and fascinatingly, almost all of them died. But what was really surprising, we put a lot of effort into designing this really beautiful experiment, and it turned out the whole thing was a waste because They all died across all of the experimental treatments, and it had nothing to do with Scotch broom competition. As you can see in this picture, there's no Scotch broom and there's a dead Douglas fir, and that's pretty much what we saw. So what was really interesting about that is that it wasn't about the competition with the Scotch broom, but the Scotch broom, by coming into these clear cuts, had changed something about the soil that made it so these areas that used to support forest could no longer support Douglas fir. And then tromping around up there in a lot of clear cuts, one of the things that I noticed, so these are old clear cuts that had been sort of planted where they removed the Scotch broom, planted Douglas fir again. Scotch broom grew up, no trees, remove the Scotch broom, start over again, like 2 or 3 times they'd had failures in reforestation. But when I walked around in these clear cuts, what I noticed was that if you look at the edges of the forest, there was always like a row of trees from that planting that had made it, but out in the middle of the clear cut, there was nothing. So that got us sort of thinking about what might be going on in the dynamics. And that got us thinking about the role of mycorrhizae. So I had a wonderful PhD student, Sarah Grove, and a cool undergrad who was a double major in plant science and chemistry, Jennifer Thompson. And we did a whole bunch of studies. We started getting really interested in what goes on with the interactions between the Douglas fir and their mycorrhizal fungi. And we did a bunch of experiments where we were culturing mycorrhizae off of Douglas fir roots and then looking to see how the chemistry of Scotch broom was affecting their growth. So in addition to a bunch of greenhouse experiments and other things, one of the things that Jennifer did was look at just how the alkaloids inside of Scotch broom leaves. So alkaloids are things like nicotine and caffeine, this group of chemicals that have antimicrobial properties as well as can affect herbivory. So we did these trials where we looked at fungal growth at different concentrations of aspartame, these alkaloids out of Scotch broom, and we found this very strong effect. So it looked like the Scotch broom was actually poisoning the soil in ways that reduced mycorrhizal colonization of the trees. So we did a field experiment next where we were trying to look at how tree seedlings would come back, or what their success would be close to the edge and further out into the clear cut. And we attempted to do an experiment to see, well, hey, now that we've figured out that soil seems to be a really important part of this, could we just fix the situation by dumping literally a yogurt container of soil from the forests where we had these wild, very rich mycorrhizal communities into the hole with the seedlings. So we planted about 1,000 trees across 5 different sites, and we had them set up along these transects where they were either close to the edge or farther away. And then they either got soil collected from these mature Douglas fir forests or from the surrounding clear-cut cytisus, that is Scotch broom invaded soils. So now I'm going to show you some of the data from that study. So in terms of the effect of edges, it was much better for the seedlings that got planted right along the edge of the forest. So if you look at these 2 graphs, see if I can get this to work. On the bottom panel, we've got mycorrhizal colonization of the roots. And on the top panel, this is survival of the tree seedlings. And on the left-hand side, these are the ones that were planted out into the middle of the clear cut, the far ones, and the near ones, the edge tree seedlings. So it was always better to be on the edge. And the ones along the edge were colonized by a lot more mycorrhizal fungi. So the fungi were supporting the growth of the tree seedlings, and that was happening along these forest edges. In terms of the question of like, well, so was that the key to restoring forests? Unfortunately, the answer there was no, because if you look now at the yellow and green pairs of bars, the yellow ones are the ones that got the yogurt container of Scotch broom invaded clear-cut soil. The green ones got the mycorrhizal soil from the Douglas fir forest. They didn't do better. These data are from 18 months after the experimental treatments were done. What was disappointing about this was that when we collected data at 6 months, they actually did show a significant difference in that it looked like they were going to do better with the addition of mycorrhizae. And another interesting thing from this study was that we did find lasting differences in the fungal communities on the roots of the seedlings. So we were able to shift the fungal communities in a way that lasted. So when we used DNA sequencing to figure out which were the fungi on the different seedlings, we found that fungi from the forest did colonize the roots of the plants that we added them to, including a couple of really what we know of as important mycorrhizal species like Xenococcus. So if you look at this one, the green bars are way higher. There's much more abundance of them, those particular mycorrhizae, but it didn't have an overall effect on the plants. Sad. Why? Why didn't that work? In thinking about various options, possible explanations, one of the things that became clear is that mycorrhizae, you can't always just pick them up and stick them in a hole. And around this time was when people started really talking about the importance of mycorrhizal networks. So we're going to talk about that next. All right, what are mycorrhizae? How do they work? Mycorrhizae are a mutualism between a plant and a fungus, where the plant, which is photosynthesizing, so it's creating carbohydrates using the energy of the sun. Fungi can't do that. So the plant is generating these carbohydrates and sharing them with the fungus. And the fungus, in turn, is providing the plant with nutrients. Why does the plant need the fungus for nutrients? Fungi have these incredibly thin hair-like hyphae that go out into the soil. So fungal hypha is so much smaller than a root that it can access different parts of the soil. So that means that mycorrhizae can be important for not just nitrogen and phosphorus, but also water sometimes. So they can be important for different things in different contexts. Some fungi can also do really cool things, like some fungi have enzymes that allow them to access certain types of phosphorus that are not available to plants. So that's another thing that in some cases are really important. So we've known about mycorrhizae for a long time. They were first described in 1885, but more recently, people started realizing that this idea of a plant paired with a fungus is actually not how many mycorrhizal systems work. So in many cases, mycorrhizae are actually forming this incredible complex network below ground where they are spreading all over the place and connecting with multiple individuals of plants, sometimes just one species. Sometimes they'll actually be in association with multiple species in the same mycorrhizal network. So multiple fungi, multiple plants, all connected underground, all sorts of stuff happening. And at first, the first experiments showed that these mycorrhizal networks were ways in which the fungi weren't just directly interacting with providing nutrients to one plant, but could actually move nutrients from one plant, or even one plant species, to another one. So now you've got carbon moving between plants. And it was people like Joe Emeriti at University of Washington who said, like, these mycorrhizal networks can be huge. So if he found what he believed was a single fungal individual that was like acres large. And then people like Suzanne Simard came around and discovered that not only are nutrients moving in these mycorrhizal networks, but also information, so that the plants were sending messages to each other. So I want to unpack that a little bit so we can kind of get a better understanding of what we mean when we say plants are sharing information. Okay, so plants have they have a whole series of signaling compounds, but they're it's a pretty manageable set. This is not like, they're not sending text messages to each other, right? Like there are a set of signaling compounds that are involved in this kind of thing. One group of them are plant growth regulators. So you've got plant growth regulators that are things like auxins, which, by the way, the chemical structure of auxins were first described by Kenneth Tiemann, our very own faculty member, and cytokinins and gibberellins. So a set group of compounds that help the plant figure out which end is up and how to grow and where to make branches and that kind of thing. Important to know that fungi actually have figured out how to manipulate plants, and in some cases, will actually produce these plant growth regulators themselves. So the way that gibberellins were first discovered was by a Japanese scientist who was studying a disease of seedlings, rice seedlings, that causes he called it foolish seedling disease, because it causes seedlings to grow all skinny and then fall over and die. And so by doing that, he figured out what these gibberellins were, and then they discovered that gibberellins were generally produced by plants for their own functions. So anyway, so that leaves open this other interesting question about how the fungi in the plants are interacting in ways that might not be to the plant's benefit. Okay, then the other group of signaling compounds that are important in this context have to do with resistance, plant resistance. So plants don't have immune systems like we do. They have but they can have systemic resistance. So a plant that gets infected, where one leaf gets infected by a fungus, will upregulate a whole bunch of defensive mechanisms within that leaf. And then they'll also send a signal through the plant body so that other leaves will also upregulate their defensive mechanisms. There's basically three signal compounds that are involved in that: jasmonic acid, salicylic acid, and ethylene. Jasmonic acid and salicylic acid are involved in slightly different types of fungal infections. And jasmonic acid is also involved in resistance to ovivary. So those are the kinds of things that people are finding moving from plant to plant. So rather than it being like a text message, it's more like somebody's car alarm just went off on the other side of your neighborhood, and you're trying to figure out now what's cool and super complex is how do the plants and plant tissues figure out how to interpret those signals. And it all depends on context and the cells and what all is going on, where they are in the plant body, and all of these things, like how the plant is actually responding to those signals. Okay, so that's what we mean when we say that information is flowing around. What has been super interesting to me, and for the plant biologists in the audience, has been, so when I started working on mycorrhizae, this was like niche field. Like nobody knew about this stuff. It was like the super nerdy, very esoteric thing, kind of like working on lichens, Laurie. And then suddenly it's just been so interesting to see how this has kind of exploded on the scene of the national consciousness, and how many interesting books for the public are out there now exploring what fungi do and the interactions between plants and fungi. And as well as some super interesting, fun fictionalizations of that as well. So I'm a huge fan of magical realism and sci-fi applications of crazy ideas about how mycorrhizal networks work. So I love Avatar, and I've read the Overstory twice. So I'm not at all in any way sort of against exploring the cool possibilities of what fungi do. But for those who work on mycorrhizae, this has been also kind of a little bit of a fraught moment because of the way that this information has sort of gotten out there and the kind of conversations that people have about it. So this is something that I just pulled off the internet about this idea of the mother tree, which was a term that was started by Suzanne Simard, whose research I mentioned earlier. that the mother trees can pass on information for best growth patterns and can divert nutrients to trees in need. So all of the ideas you might have in your mind about what it could mean to be a mother tree, that's what these conversations are about. So it's really this concept of trees as being very altruistic and that there's a communal life in the forest. And interestingly, that concept is sort of in tension with a different way of thinking about the interactions between plants and fungi, which has more to do with individuality and the fact that each organism has its needs that it's trying to meet. And so sometimes those needs are in sync, sometimes they might be in antagonism. So I think this is a really nice article in the New Yorker from several years ago. The Secrets of the Wood Wide Web, which is a term you see. And I'm just going to read it because I think it's really nicely stated. A central debate over the Wood Wide Web concerns the language used to describe the transactions it enables, which suggests two competing visions of the network. The socialist forest, in which trees act as caregivers to one another, with the well-off supporting the needy, and the capitalist forest, in which all entities are acting out of self-interest within a competitive system. So the sort of standard bearer of the capitalist forest concept is a really wonderful scientist named Toby Kears, who has done a lot of really interesting work. And her favorite metaphor for this is the underground market. And this is another article from Quanta magazine. Kears, a benevolent cooperation focused view promoted by thinkers like Margulis and Samar to suspect. She saw instead a world ordered by individual interests where potential cheaters lurked everywhere and species needed complex strategies to keep their trading partners in line. And her science is so interesting because she's used a bunch of really cool, innovative approaches to understand the natural history of what these fungi are doing underground. And one of the things she does is she has this quantum dots approach where she tracks individual phosphorus molecules or carbon molecules, and she actually looks at what's going on between an individual fungal hypha and root, and under what conditions do they cooperate, and when do they withhold their resources, and it's all super interesting. Okay, so this is the point where we're going to have a discussion. And I want to say that I am not a critical theorist, and I'm also only a little bit of a socialist. And so I try to be careful about the sort of my own interpretation, but I guess to be fully transparent, I should share that I would say most plant biologists, and certainly in the ones in the table to my right here, who have actually published on this stuff, not me, find the evidence for this idea of individuality more compelling. And when I say that, what I mean is that the implications of it or the sort of the way it works feels more in line with the way we understand how natural selection and evolution work in organisms and in coevolutionary dynamics. And then also one other thing that I think is relevant is that when Suzanne Simard has been kind of pressed by the biological community, she has herself said that she thinks of the mother tree idea as more of a metaphor as opposed to a description of scientific reality. And so I thought, knowing the amazing range of big brains in this room, that it would be really fun to have you discuss this prompt, and I'm just going to walk around and listen to all the wisdom being shared. So here's a question. What do you see as the relationship between science, metaphor, and truth in this conversation about the Wood Wide Web, although you may have experience with this kind of question in other spheres or other contexts. So I think it's fantastic that our Emeritis go across all of these fields. There's so much experience in this room, and I think this would be a fun conversation to have. So what we're going to do now is I'm going to give you 10 minutes to discuss. I recommend groups of maybe 4. Don't let the groups get too big so everyone can express their thoughts. And when we come back, we'll just completely change topics. So don't hurt each other. But yeah, 10 minutes. So I'll give you a 1-minute warning when we're getting done.
What's the answer?
You see how I changed the slide?
How do you get on the fire when you started?
I knew this would happen. I knew what I was up against. Okay, everyone, that was awesome. And I can't wait to follow up with people to hear about what the conversations were like at your table. Frank just asked me what the answer was, and I loved it. Okay, so I do think it's really, really interesting, and I want to reiterate that regardless of how you think about exactly what metaphors we use or exactly what is the nature of these interactions, that they are amazing and complex and fascinating, and we don't understand even a small proportion of what's going on yet. So I think that's really exciting. So now what we're going to do is talk about fungi in another realm, which hasn't really hit the popular press yet, but is equally amazing and cool. And that is the fungi that live inside of leaves. So we use this term endophytes for species that live inside of leaves, endophyte. And leaves are full of fungi. I mean, other bacteria too, but especially fungi, many different species, and they're doing all sorts of different things. And one of the things I love about working at, so first I'll mention that the rest of the work I'm going to talk about today was all done in collaboration with my husband, Greg Gilbert, who is a professor of environmental studies, and bless his heart, he's also the chair of CAP this year. Everybody knows him. And he will be retiring in June. So he's going on to bang. Maybe he'll be with you guys next year. But one of the things that's really fun about working on endophytes is that one of the first people to describe endophytic fungi in leaves was Jean Langenheim, who published a wonderful paper with her PhD student Francisco Espinosa Garcia in 1990, where they described communities of fungi living inside of regular leaves. So thanks, Jean. So fungi inside of leaves are doing a lot of different things, which I'll talk about in a bit. But I think it's important to get out on the table that one of the important things that fungi do is they cause infectious disease. So unlike in humans, most plant disease is actually caused by fungi. Other microbes are also involved, but it's mostly fungi. And this is a topic that I've been thinking about a lot. Greg and I have been working on this for a number of years, and we have a book that came out in Oxford Press a couple of years ago now, exploring these ideas. So it's definitely near and dear to my heart. My first exposure to plant endophytes was studying plovers up at Bodega Bay. So if you've ever had a chance to go up the coast, the Bodega Bay Marine Reserve or the state park up there, this is a beautiful area. And one of the awesome things about the Bodega Marine Reserve is it's full of clovers, but you didn't know that. Clovers have a global hotspot here in California. So there are actually 33 different species of native clover across our state. And then there are all these clovers that people brought here because they also are a good forage for animals and stuff. So Greg and I started working on the interactions between clovers and the fungi that live inside of them because we were interested in comparing native and non-native species, but I'm not even going to talk about that today. Back then, we didn't have DNA methods for identifying what was going on inside of leaves. So we were doing a lot of culturing. And we got really interested in this one particular fungus that was infecting all sorts of clovers, Stemphylium solani. So we spent a lot of time understanding really getting into the nitty-gritty and the mechanisms of this interaction between Stemphylium solani and plants. So one of the things we did was these enormous greenhouse experiments. So thank goodness we have amazing greenhouse support here. at UC Santa Cruz, and we were growing thousands of plants and tying threads around individual leaves, like 6,000 individual leaves, and then infecting them with Stemphylium solani and looking at how it affected plant fitness. So it has this effect of leaf necrosis and chlorosis. It causes early senescence of leaves. And that makes sense. I should have said this before, because the way that this fungus reproduces is by growing on dead plant tissue. Okay, so it needs to kill the plant tissue first, and then it can reproduce. Okay, so one of the things we did was look at the evolution of this pathogen and how after infecting the same species for 5 generations, we saw this really rapid evolution of the ability to infect that particular host. So we were really interested in the dynamics and evolution of host specificity and coevolutionary dynamics. And then we did these big field experiments. You can see all of our flags there, where we planted out clovers, little tiny clovers at each of those flags. And then we tracked what happened with their interactions with fungi, fungal pathogens, as well as herbivores. So one of the things we did was a big fungicide experiment where we sprayed the plants every other week, and we got reductions in the infection rates. And then we looked to see what happened. So as you might expect for a pathogen, when we spray these plants with fungicide, we saw the mortality rates drop. Then we did the experiment the next year. Dun, da, da, and we saw the opposite thing, which was wild. So we reduced infection on the plants, and we found that they actually did worse. So, and this was with a fungus that we had already, several years of working with. So we knew that under certain circumstances it was causing leaf senescence and all that stuff. But sometimes having fungi in your leaves was actually beneficial. And that was really, really cool. And then when I started kind of getting into the literature and talking with other plant physiologists, I found folks from agriculture who've been studying this, showing that fungi inside of leaves can do all sorts of different things. But one of the things that they can do is help the plant moderate its water balance. So there's just a lot of really cool things that they do. Okay, so the take-home message there is that leaves are full of fungi. Sometimes they're causing disease. Sometimes they're helping the plants, and they can evolve rapidly. So that's kind of just a little nugget to blow your mind, so that when you look out at a plant community, you can think about all of those cool fungi that are in there doing all sorts of things. So the last piece of the story is to talk about the work that we're doing now. So this is ongoing work. It's still unpublished, and it's also collaborative with Greg. And what we decided was it wasn't enough to study 17 species of clovers and all of their fungi. We really wanted to understand plant fungal interactions at the whole ecosystem level. And so that's what we are doing now. And it used to be that when you studied disease, host pathogen interactions, mostly you studied one host, usually humans, but maybe an animal or a plant of economic importance, and one pathogen that caused disease on that host. And then one of the great things about the COVID pandemic was that it really opened people's eyes to the fact that if you're studying just one host and one pathogen, you were potentially missing a lot of things that could be important in terms of causing disease on your host. So now we know that, or people have known this, but now we're much more aware of the fact that a lot of our pathogens we share with other species, and that this movement between host species can actually be really important in the dynamics of disease. Whoops. So that's what we study in plants. And so if you want to predict disease on this clover, you need to know not just about the host dynamics of that specific species, but also its community context. So if species are sharing their pathogens with other species, that leads to all these really interesting questions about what is the host range of most pathogens? Like, are most of them specialists? Are most of them like super generalists? And which pathogens are being shared with which hosts, and can we predict that? So you can think of hosts and pathogens as communities that are in complex networks. And one of the fun things in this arena is that we've borrowed network theory which was first originally developed to understand human communities and how interactions work between humans. Now ecologists have used those approaches, those analytical approaches, to ask all sorts of cool questions about natural communities. And one of those applications is studying plants and pathogens. So if you think about the network, you can have some pathogens that are host specialists and are only found on a single host. You can have other ones that are generalists. And similarly, some hosts might have lots of pathogens that they're dealing with, and other hosts might only have a few. So in case you're interested in what these networks look like, this is the network picture for that clover system I was telling you about. So here we've got 17 different species of clovers that we studied. And here in the blue squares are these fungal pathogens that we isolated from them. And you can see that there are this is Stemphylium. So Stemphylium was found on most of the hosts. It was a broad generalist within that clade. But then you had other fungi that we only found ever on single hosts or a couple hosts. So that's kind of what the network structure can look like. When you get more complicated, like you want to look at all the species, it's easier to look at these structures in these starbursts, which I'll show you in a minute. So that's what those look like. For this project, now we get to my backyard. For this project, we took advantage of this spectacular campus that we have and its amazing ecological diversity. And we compared 3 very different ecosystems. The mixed evergreen forest of upper campus, the Great Meadow, which was just described to me as, what did you call it?
Coyote brush.
The coyote brush invading the Great Meadow, the Spanish barnyard. Somebody was just saying that was what they heard it described as. And actually, this is funny because James brought this up last year at one of these that I got to see. It's mostly our Great Meadow is mostly non-native species. Sorry, it is beautiful. It does have poppies. They're native, so that's good. And then we've got this amazing working organic farm right here on our campus. And that allowed us to compare these 3 ecosystems that are very different in their structure. If you just picture yourself in each of these places that you know well, you can think of all the ways in which these environments are different. They also differed in their species diversity of plants. So we were looking at the understory of the forest, which was like, 3 times more species at the farm when you include the crops and all the other stuff. So what we wanted to do was look at all of the species for which we could find at least 5 individuals, and then all of the fungi that were growing inside of those plants. So what we did was we used DNA barcoding methods for this. So we combined the individuals of each of the species of plants. We amplified up the DNA that was in there, so all of the fungal DNA. And then what you end up with is like this big grab bag of DNA sequences. And then you use bioinformatics to figure out each of the unique sequences, what is it? So, so that's how we figured out what all the fungi were growing inside those leaves. And this is work that is collaborative with our technician at the time, Asa Conover, who's now a PhD student at Berkeley. So this is what those networks look like. So the blue dots are the pathogens, the green dots are the plants, and all 3 of these very different plant communities have pretty similar structures. So around the outside, you have all these things that are host specialists found on only a single plant, and then you've got this kind of big cluster in the middle of plants and fungi that have lots of interactions with each other. The first question that we asked with these data is about habitat specificity. So you think about those 3 different sites ecosystems, think about how much overlap there is in terms of which plants are where. And that's this Venn diagram on the left. So each of the habitat types has its own color. And you can see that in total, 20% of the plant species were found in more than one habitat. Now, what's really interesting here is this is what it looks like for the pathogens. So the pathogens were actually much less habitat specific than their plant hosts. So pathogens are moving across our landscape and they're infecting hosts in multiple places, even in habitats that seem really different from each other. That's important if you, say, are a farmer and you want to know, where your crop pathogens might be coming from. And so we have to think about the landscape context when we think about these interactions between plants and fungi. In terms of specialization, we were asking this question about across an entire ecosystem of interactions, how specialized do these pairings tend to be? And as far as I know, ours is like the largest scale study to ever attempt this. I don't know of any other examples where people have tried to look at host specificity across all of the species in the community. And this is what we got. So the majority of pathogens, so this is the number, so this is a histogram like a grade distribution, where on the x-axis is the number of host plant species. So this bar would be the ones that were only found on a single plant host. And this bar would be the ones that were found on 8 to 15 plant hosts. And this whole thing together shows that most of the fungi are found in multiple hosts. But there are a pretty big group, a third to a half, that are more host specific. And that brings us to probably the most important question, which is which of species are sharing pathogens with which other species? So we've known for a long time that closely related plants are more likely to share pathogens than our distant relatives. So if you have a tomato in your garden, it's more likely to share pathogens with potato, which is in the same genus, than with your broccoli plant, which is in a totally different plant family. So that's something we know, and the reason behind it is because closely related plants are more likely to share traits that then affect how they interact with microbes. So the fact that more close relatives share traits is something we call phylogenetic signal, and we can quantify that. So if you think of the plants in your plant community as kind of being arrayed on a phylogenetic tree, like your two grasses are more closely related to each other, your two legume plants, your clover and your lupin are more closely related to each other than they are to other species. So you can lay out all of your plants on a phylogenetic tree, and that allows you to calculate phylogenetic distance between any two species in your community. And that's a really cool tool. And then you can use that phylogenetic distance to predict, in our case, which species are most likely to donate their pathogens to the focal plant that you're interested in. So what we would predict based on those things is that plants in a plant community that are phylogenetically isolated, meaning they don't have any close relatives, should have fewer pathogens than species with many close relatives. And that's actually exactly what we found. So each of these dots is a species in the plant community. And we're calculating here the 10th quantile distance between that focal species and all the other species. And what we find is we can predict this relationship with the number of pathogens that plant is associated with. Part of why that's really interesting to us is that a number of years ago, we did a study where we were able to predict, we were able to show that plants that were more phylogenetically isolated in that way that I just mentioned showed less disease. So that's a log scale. So if you just look at the effect of disease, or what we call disease pressure on a species, it is predicted by its phylogenetic rarity. Okay. And this has an important application. So one of the things we care about is the health of our cropping systems. And you might expect, you would predict that pathogen sharing, so two species, the probability that they share a pathogen should decline with the phylogenetic relatedness between the crop and the other species around it. And our data set allowed us to study that. So we were able to look at, because of the amazing diversity at our farm, we could look at 34 different crop species, annuals and perennials, deciduous and evergreen. And in all of those 34 cases, except for one, we saw this very strong confirmation of the predicted pattern that crops were more likely to share pathogens with their close relatives. And what's interesting is they're likely to share with their close relatives in these, even in these distant plant communities that are far away from them. That had important implications for cropping practices like intercropping or crop rotation. So if you think about agroecology and the design of a farm, it also has important implications for spillover of diseases from our agricultural crops into our wildlands. So both the work that we did on habitat specificity as well as this work really points to certain types of risks that we might have by amplifying up plant diseases in agricultural systems that then can spill over into wild systems. And for those of you who are in the anthropology world, one of the things I love is that our traditional ecological knowledge and traditional cropping systems actually fit nicely within this framework as well, because traditional cropping practices like our three sisters, maize, beans, and squash being grown together, clearly fits in. These are three species that are very phylogenetically distant from each other. And so you would expect that a design like this would naturally suppress disease in your cropping system. Okay, so that's the end. In terms of fungi inside of roots, mycorrhizae mediate interactions between plants. Unfortunately, it's hard to restore them in disturbed sites, but there's so much more to learn about mycorrhizal networks and how they work. Fungi inside of leaves, they're everywhere. Their interactions can shift between mutualistic and parasitic, and context matters. And then you can broaden that out to study whole networks of plants and fungi where your phylogeny structures disease interactions, and we can predict some things, which is great. And I'll just leave you with the idea that pathogens are natural parts of ecosystems. They can help maintain diversity, and they're very worthy of study. So with that, I'll thank everyone, this great university, all of the people who support our research, funders, and my collaborators, and thank you for your time.
Well, thank you so much, Ingrid. Unfortunately, that break used up our official question time. So if anyone has to leave, this is a good time to leave. But I will allow time for questions, but
Feel free to go.
So I noticed that your experiment planted the same kinds of trees, but I recently read an article about these little tiny gardening centers planting lots and lots of species in urban areas, and they're private. So how does that fit into your research?
Yeah, well, those earlier experiments were with Douglas fir. Repeat the question. Thank you. Yeah. So the question is, in forestry systems where you're planting one giant monoculture, all of one species, there are some efforts now, especially in urban forestry, where they're packing lots and lots of species into small areas and having a lot of success with that. So I think in that case, you're kind of using ecology in a positive way, because we know that there's lots of benefits to having species diversity in a system. And it's not surprising that giant forestry systems like Douglas fir timber production in the Northwest has certain kinds of, it's going to have some limitations. That said, Douglas fir forest that naturally is actually it's not a complete monoculture, but it's not very diverse in terms of the trees. It's pretty diverse in the understory. So there are reasons why timber production is kind of a natural fit with that ecosystem. But I love that you brought that up, that example, that there are these urban areas. I've read those studies too, where they're like just putting tons of species together, and it increases the total resource use, and it probably also reduces disease because of the way that disease, there's a thing called the dilution effect, which is that you can suppress disease in a community sometimes by increasing the diversity of the potential hosts. Yeah, Lori.
Thank you so much for that talk. I just wanted clarification. When you were talking about pathogens in the third section, were you referring to all the fungi you were finding, or were you referring only to the parasitic destructive fungi?
Great question. We have a data set that's all of the fungi, but all of the data that I presented today was just the parasitic ones. And the way we figured that out was by using, so there are these cool databases of mycologists across the world who have curated databases that on the functions of different fungal groups. So there's like ones where we don't know anything about what they're doing. We didn't include those in that subset of the data. And also the ones that are known to be more mutualistic. So there's a whole other paper to write on the other groups of fungi. And the ones that I showed today were just the 390 species that are known pathogens. Thanks. Yeah.
I have a question about the Scotch broom versus forest portion of your talk. Were the new trees that did grow at the edge of the forest only growing because of the seedlings, or was there any sign that the forest itself, through its natural dispersion of seeds, was producing more forest?
Oh, great question. The trees that I'm talking about were planted in the edge, and we did some experiments too where we planted tree seedlings and followed their success. But you're totally right in that you might expect some natural regeneration to happen as well. And another aspect of it is there's also other reasons why you might expect the edges to be better for trees. Don't have to do with mycorrhizae, like they're slightly more music, they're a little moister because they're not in the direct sun, that kind of thing.
Follow-up. Does that mean that over the centuries, the forest would gradually creep across the Scotch broom? I'm an optimist, sorry.
Yeah, no, that's debated. That is debated whether the Scotch broom is a permanent stable state or if it's a transitional state. And what I have observed, it's hard for me to believe that those trees are actually encroaching, but with some help. So what we ended up saying to our forester collaborators was, sorry, there's not an easy fix here, but in the future, please make clear cuts very skinny and narrow, like long and skinny, because the more edge there is, the better chance you have of getting your trees to grow. And so what I'm hoping is like these big clear cuts are really a sticky problem, but I'm hoping that they can do it kind of one row at a time if they think long term.
Thank you.
David, question.
Yeah, hey, Dave.
Wondering whether your pathogenic fungi themselves have set of enemies, what they might be, would that be another factor you brought in?
Funny you should have asked that question. So David's question was, do the fungi also have enemies? And so is there sort of a even more complex web going on? Greg and I have a PhD student right now, Edith Lai, who's gotten very interested in powdery mildew, which if you walk around the forest, you probably see it on Stachy's gulata and some other plants. Powdery mildew is that white stuff on the leaves. Powdery mildew has a fungus that eats it. So she's studying this three level interactions of this mycotrophic fungus that eats the powdery mildew fungus that has a very interesting host specialized relationship with its plants, and including grapes. So we're gonna be growing some grapes, I hope, in the future. So there are some really fun interactions like that. Yeah, totally. Are we getting to the end?
Oh.
Or
What variants?
I've read that there's been attempts to enforce some of the Scottish Highlands. Is broom interacting there with, I was wondering.
Cool question. So Scottish Highlands, and there's some interest in bringing back forest up there. And the question is there a similar interaction between broom and trees up there? I don't know about that. Broom, I have gotten to see Scotch broom in its native range. And it's not nearly, it doesn't look, when I show pictures like that, to colleagues in Europe, they're like, oh my God, like that's crazy. So it does.
It's up there.
Right. Yeah, I did have a collaboration. We tried to get some funding for studying the Scotch broom with the native Douglas fir, native Scotch broom in Europe where Douglas fir is introduced, and in New Zealand where both species are introduced. But we never got funding, so.
Yeah. Maybe one more question, there is.
Well,
Roger.
Yeah, I'm curious what is known about how these diseases are transmitted between different plants. Is it is it mitigated by various other animals, let's say, or by?
Great. Great.
Or is there some sort of a serial transmission? Or there's some plants which act like typhoid berries in a way. They are receptive to these pathogens. You can more easily pass them on.
Yeah, all of the above. So the fungi that we study, some of them are windborne, and you can pick them up by doing spore sampling. Some of them are water splashed, like the powdery mildew we were just talking about. That won't those species my student is finding, they won't go anywhere in the wind, but they have to be splashed from one plant to another. And then the typhoid Mary thing, I hope you all know about sudden oak death. And the fascinating part of the sudden oak death story, which is a species of oomycete, which has been killing our oaks, especially our tan oaks on campus. What's super interesting about that story is that the oaks can't spread sudden oak death from one, well, very rarely does it spread from one oak to another. Instead, the typhoid Mary of that story is the bay laurel. So if you have property where you have some beautiful oaks that you're trying to protect, our recommendation is you get rid of your bay laurel, because bay laurel gets infected, pumps out huge numbers of spores from its leaves, but it doesn't die. So it doesn't get infected. It doesn't produce cankers the way that oaks do. So yeah, so many cool questions. Thank you so much.
Thank you.